Hi folks, welcome back. We are in lab number 9, one more to go after this, and in this one I'm going to go over the split plot design and its relatives. So that's the split plot, the split, the split block, and the repeated measures. So let's get started. Just to remind you from lecture, all of these split plot designs, basically it comes down to the fact that because you have constraints on randomizations and multiple randomization events, and therefore different experimental units for different factors, you have different error terms for different factors. So largely the models are going to look quite similar to what we've seen before, but they will include, the only difference is that you'll be fishing error terms that you need out of the residual and putting them in the model so that you can use them. In example 1, we're going to talk about a split plot design organized as an RCBD. And just to remind you what that means in terms of randomization is that you go to the field, you define what a block is, and then you carve that block up into the number of levels of your main plot. So here we have 4 main plots, which are varieties of oats. And then the second randomization event happens. You go inside each main plot and divide it up into the number of subplot levels that you have. And in this case, we have 4 seed treatments, including 1 control. So one block, as you can see, has 16 experimental units in terms of the subplot, but only 4 experimental units in terms of the main plot. And the whole study would have 4 total blocks. So that's the layout. And let's go ahead and look at this in R. We're looking at example 1. I've loaded in my data set. It's called oats.dat, and here it is. We have block IDs, we have factor A, which is variety, and I'm putting the A in the treatment name so that we can keep track of what is a main plot and what is a subplot. So variety A means variety is my main plot factor, and we have treatment B just to designate that that is also a factor, but it's my subplot, and then our response is yield in each case. Once you bring it in, you're going to look, of course, at the structure. We see that neither block nor variety, or actually nor treatment, are being treated as factors, so that's what lines 13 through 15 do. We reassign them as factors so that we can call upon them in the model, and then we specify the model itself. Here, instead of using the LM function, as we've been doing up to this point, I'm using the AOV function, which is useful in some instances, particularly because it allows you to designate within the model one custom error term, and that's what you see right here. I'm using this error function within AOV to declare my block times A, my main plot block interaction, as an error term for all of the terms that are implicated in this interaction. So by programming it this way, I'm going to get A in my model, block in my model, and it's going to use block times A as the error term for running the f-test for those 2 factors. I'm also going to get B, A times B, and those are going to end up using my residual error term. So that's exactly what I want to do. I can define the model, ask for the summary, and you can see very nicely that R gives me an output here, in which my ANOVA table has been broken up into 2 parts. I have a top part here for the main plot effect, A and block tested by the A times block interaction, and you see here error is the A times block interaction with 9 degrees of freedom. And then below that is another part of the ANOVA table with B and A times B and using the residuals. So in terms of running the model, it's quite simple. The only difference, the only thing that distinguishes this model from a regular RCBD with a factorial treatment structure is the inclusion of this term. That's the only thing that differentiates it. Now, looking at the results here, we see that we have a significant 2-way interaction between variety and treatment. And so therefore, we are not justified in doing a main effects post hoc analysis of either factor. We need to look at the simple effects of these factors. What I have in the rest of the script here is doing exactly that. I am subsetting the data. So here, I want to compare my subplot levels, compare my treatment effects within common main plot levels. So you've seen this before, in which you take the full data set and subset it by level. So here I'm grabbing all of my data on variety A and putting it into a subset that I'm calling lot 1 data. And then I'm running a reduced RCBD. Now it's just a regular RCBD where A doesn't enter the model at all. The residual is the correct residual. And in this particular case, I'm calling on the library multcomp so I can run Dunnett's test of every treatment versus the control. And that's what you see here. And now because I have 4 varieties, I'm running this code 4 separate times. There's variety 1, 2, 3, and 4. And that's a lot of programming. So what I've also given you here at the bottom of the script is a way to do that same analysis using a loop in R. This is not something I expect you to do, but if you are interested in making your programming a little bit more efficient in R, I recommend you take a look at this, and I'm happy to talk with you if you're interested at all. Okay, so that's really it. What I want you to do, I think what we should do now is let's pull up the next example. And what this is example 1b. What we're doing in this example is I'm taking the original data set, but this time I'm averaging out the subplot entirely. So if you look back at this data set, let's see if we can make some sense about this. I have block 1, variety 1, and there are 4 subplots within that. Block 1, variety 1, treatment 1, and I go down a little bit. I have block 1, variety 1, treatment 2. I have block 1, variety 1, treatment 3, and block 1, variety 1, treatment 4. In other words, for that one combination of block and variety, if we go back up to our map here, that one experimental unit for the main plot, I'm going to have 4 subplots. From the perspective of the main plot, from the perspective of variety, those 4 subplot treatments are subsamples. And we know that with subsamples, unless you're interested in the components of variance or characterizing them, you can simply average them. And so that's what I'm doing in example 1b. If we pull up the data set for example 1b, and let's open up that R code too. So I'm going to open up example 1b, the script. I'm going to open it in RStudio. It's going here. And I'm going to bring in, I'm also calling that oats.dat, so I'm going to clear my last data set. Clear it, and I'm going to import 1b, the CSV file, and I'm going to call this oats.dat. Okay. What you'll see is that this particular data set is much, much smaller. In fact, it's a quarter of the size of the original data set, because what this data set is essentially averaging over the subsamples within each block A combination. So instead of having 4 lines in the data set with block 1, variety 1, I have 1 line, and then this yield number is the average of those 4. Under this circumstance, let's run the code. Under the circumstance, I need to get A and block as factors. B has dropped out of the experiment entirely. This is simply an RCBD with variety A as my treatment effect, as my treatment. And so we know that for an RCBD with 1 treatment factor, this is the correct model to run. And let's run that. And what you see here, which is pretty cool, is that we get the exact same results here as we got in the original analysis, in which B was also a part when we used the correct error term. So this is a confirmation to us that, first of all, A times block is the correct error term to use, because in a much simpler RCBD case, that is the error term that we use. And second of all, that we can now include the B, factor B, within the same model without at all affecting our conclusions about A. In other words, what's happening at the main plot level is insensitive to whatever is happening at this level of the experiment. And so that's what example 1b is meant to illustrate. Just another way to think about why the block A interaction is the correct error term. We also, following that, talk about a split plot design as a CRD, in which case the randomization looks quite a bit different. Rather than breaking the, originally we broke the whole field up into 4 blocks, where these 4 main plots are 1 block, this is 1 block, this is 1 block, and this is 1 block. As a CRD, you would just approach a field of 16 experimental units, and you would assign the 4 replications of the main plot at random among them. And so in this case, you can see main plot 1 and main plot 1 are right beside each other, which would not happen if you were to block first. After that, within each main plot, you again randomly assign the 4 subplot levels. In that particular case, and this is what you see in the example 2 script, the only thing that changes is that you run an otherwise CRD factorial model of A, B, and A times B, but you have to include the proper error term for the main plot A. In this case, it's variety A times rep. And I think it was brought up in class, when you do that, there are some requirements on what your data set needs to look like. So let's pull in that data set. I'll open up example 2. So this is a split plot a CRD. I'm also calling that oats.dat, so I'm going to get rid of my old data set so I don't have any conflict there. And let's bring in example 2 CSV. What you'll see here is that in order for replication, in order for us to be able to call up rep in the model, rep has to exist as a classification variable in our data set. And so, therefore, in order to do this, there has to be a column out here. If you noticed before, this column was a block column. Here, I'm calling it, this is my rep column. And so I have rep, A and B, and the reason rep is there is because I need the A times rep error term pulled out of the residual error so that I can use it on A. Just as in an RCBD split plot, the only thing differentiating the linear model from a normal RCBD factorial was the inclusion of the A times block error term. In this case, the only thing differentiating this model from a straight CRD factorial is the inclusion of the A rep error. Otherwise, everything else is the same. If you were to run this model, let's make sure, let's set up all of our factors. Again, rep, I'm making sure R understands that rep is a factor that I'm going to be calling upon, so that when I run the model, what we see is we have variety A. We no longer have significance of variety A. If you remember back in the RCBD case, we'll scroll up here, variety A was highly significant. And now we see that under a CRD, we lose significance. And this makes some amount of sense because block was explaining some amount of the variation in our experiment. When we lose that explanatory factor of block, all that variation goes into the error term and makes our test for variety A less significant. So it's not surprising that we lost significance there. But the main point to take home here is that variety times rep was used correctly as the error term. Following in the script are the same Dunnett multiple comparisons of each of the subplot treatments within each common main plot level. And of course you could subset the other way. You could look at main plot effects within each subplot level as we discussed in class. And this is what those same statements would look like within a loop. Now in the end, what all that is going to do is going to spit out an awful lot of output, which you would then need to summarize here, I think, on page 3 of the lab. This is an example of how you might go about summarizing the data that you get from all those different ANOVAs from the simple effects analysis. So I think that is about it. On page, let's scroll down here, at the bottom of page 6, I also have a schematic that shows you how the same split plot randomization would happen within a Latin square. And in that particular case, the only thing that changes relative to a standard Latin square linear model is the inclusion of the A times block error term. In this case, we have 2 block variables, so it's row times column times A is the correct error term. So that's it. I've hit my 15-minute limit, and we'll start with the next video.